So there is an NPTEL course also both on plant cell based bioprocessing as well as bioreactor design and analysis. I was mentioning to some of the team members here yesterday. So those who have their interest ignited after the course and want to get into more details, please feel free to enroll yourself for the NPTEL courses to know the details. Now, I already had mentioned to you, there are many examples of commercial entities, both working with plant cell fermentation as well as hairy root cultivation. Now, it is very important. Yesterday, during my conversation with you, I mentioned that it is important to note what is the difference between microbial cells and plant cells, so that when you are adapting the microbial fermenters, whose design is well established in the industry, for plant cell cultivations, you will be able to optimize the design parameters well. So if you see the size, plant cells are relatively much larger in size than the microbial cells. This will have an impact on the shear forces which get generated, the impact of shear on them, the composition of the cell wall. Like for example, in your plant cells, yesterday there was a question that they have a thick cell wall, a well-defined cell wall, then why shear? It's because of the high tensile strength and the cell wall rigidity of plant cells that they become more shear sensitive because they are less flexible. The homogeneous suspensions, microbial cell suspensions, if you will see, they are very fine. You don't have to worry about the uniformity, the homogeneity. Once you will create your cell suspensions in shake flask, many a times depending on the water activity in the callus lines, which means the fresh weight to dry weight ratio of the callus, which is very important. Although a trivial point it may seem, but it has an implication on how well is your suspension developed. So generally what happens that I'll also talk about one of the challenges we faced in the lab. I was talking about Helianthus annus metabolic engineering, and we were able to achieve 10-fold high alpha tocopherol yielding cell line. But then at the same time, what happened that the callus, though was giving us 10-fold high alpha tocopherol yield, it became very compact in nature. Compact means the water activity reduced. The fresh weight to dry weight ratio reduced, which means the friability reduced. Now, what is the issue there? The issue became bringing it to cell suspension cultures and getting a homogeneous suspension. And unless you get a relatively homogeneous uniform cell suspension, it becomes a challenge to cultivate them and scale them up in the reactors. Scale up becomes a challenge. Optimization in the reactor would become a challenge because then you would have mass transfer limitations. There'll be cell settlement at the bottom. So they become heavier. So all these will eventually then end up giving you lesser productivities. So every time it's a balance. You may be able to see how you were expecting you have got the result, but some other thing which you had not anticipated might be becoming a hindrance in your bioprocess. So cells, as I said, plant cells, they tend to aggregate, and you may be able to see as large as 2 millimeters diameters of cell aggregates. Now, growth rates we know your microbial fermentations generally get over in hours, 24 hours at the max. Now, fungal, a bit more. But with plant cells, anywhere between 12 to 15 days would be your batch cultivation period. And further, if you want to do high cell densities, you would try to do fed batch cultivation. It further increases to 20-25 days. And then possibly if you want to do continuous cultivations, which is actually a very practical challenge with plant cell suspensions, also looking at the sterility and long drawn fermentations, this may go even to months. Shear stress tolerance. Now, the same thing which I mentioned, because of the rigidity, the high tensile strength, they may be less shear tolerant. And also because if the cells are more matured, they will have an extended vacuole. This also makes them sensitive towards any change in the medium in terms of their osmolarity, the osmotic stress. There is a good chance that if a damage happens, the cell would lyse. Aeration requirements. Although I was telling that plant cells tend to aggregate, but in some way it is good in terms of reactor cultivations, their aeration requirements are low. Why so? Because the specific oxygen demand of plant cells is relatively much less than microbial cells. Can someone tell me why? What can be the reason? What does it indicate? Because they are metabolically slow. And that's why they're doubling time difference. So because they are metabolically relatively slower, so their oxygen demands are slower. So the aeration requirements are not as high as microbial fermentations. Cultivation time, as I told you, here you are talking about weeks and months, there it's in hours. So what implication will it have in your reactor choice of reactor design? You will have to look into long drawn fermentation, the sterility maintenance. Although on paper in a lab, you may say that, okay, a complicated design may seem to be working well. Let's say with high aspect ratios, a photobioreactor, and you have made all sorts of changes in the geometrical design to improve your mass transfer, but always keep in mind the scalability. So if it becomes large production scale reactors like 200,000 liter reactors, if it is a very high aspect ratio and a photobioreactor, everything will become a challenge. So make note of the long drawn fermentations, the sterility requirements, the making of the reactor, the cleaning of the reactor, how will you harvest the biomass, how will the cleaning happen, how will you again and re-sterilize it. So the ease has to be looked into when you are fine, because once you have finalized the process at the lab scale, at the pre-pilot scale, you cannot keep changing. So everything has to be designed keeping in mind that you are going to have it at the production scale. Mostly because we are dealing with high value secondary metabolites, most of these will be intracellular compounds, wherein even if you use cell permeability enhancers, generally you will not be able to get all 100%, 90% product out. But maybe you will be able to get some percentage of product out such that you can have improved productivities if you convert them into continuous systems. So I was talking about, based on these differences, what implications it may have on the reactor design. The lower oxygen demand than microbial cultures due to lower metabolic rate means that, because there can be less aeration requirements or mixing requirements, but also please keep in mind that plant cell cultures, they show non-Newtonian behavior as they will keep increasing in density. We had ourselves done that experiment in Vaila Udarata cell suspension cultures, and we came up with a power law model where we could easily see, after doing in the log phase, that as the cell density was increasing, the Newtonian behavior was lost. So because of that, whatever models and whatever assumptions you make in your models, you assume that these are homogeneous suspensions. So therefore, there's no heterogeneity. The viscosity is always same. So you are assuming, based on these assumptions, all these microbial fermenters and fermentations have been designed. Now plant cells will deviate. What we observed, that despite the fact that we had done all the optimization at the reactor level with such high cell densities, we were around 900 grams of fresh weight in nearly 2.4 liters of the medium. By the end, the cells had settled in the reactor. Now at the bottom, now even though the mathematical model predicted that we would reach to 39 grams per liter, but what was the assumption there? We did not take into account the non-Newtonian behavior. So viscosity for us was the same. So now, because of this hydrodynamic change in the environment, there were mass transfer limitations also, and the cells settled down. So what we could achieve in experimentally with the same fed batch strategy, the model predicted 39 grams per liter, but we were around 31. Although we were happy because from the batch we had improved a lot from 22 to 31, which is appreciable. But still, the limitation was we were not able to match just because the assumptions were failing, what we had begin with. More shear sensitive than microbial cells. So cultivation has to be brought in relatively lower shear conditions. So generally, if you'll see microbial fermenters, Most common type of impellers which you will come across is Ruston turbine, because the requirements are mixing. The large amount of shear force is generated, there's a good dispersion of the bubbles, aeration requirements are met, there's a good throw till the end. So there is a good chance that there will be less dead zones. Now, what happens that if we use Ruston turbine type impellers with plant cell cultivations, you may end up in cell viability loss because of high shear forces generated. So your biomass concentrations may not match what you had seen in the shake flask. Generally, the difference which you'll find is that in microbial cell fermentations, when you'll do it in the reactors, the concentrations do improve. Why? Because we assume that reactor is giving a controlled environment of the optimum conditions vis-a-vis the shake flask, where some of the conditions cannot be controlled. So generally you will see improvements. But in plant cell cultivation, sometimes what may happen, you may see that not only that it has not been able to match, rather it will dip from the shake flask. Some of the reasons can be this as well. The shear forces, the choice of the impeller, which may have hampered the cell viability. So therefore, we make use of low shear generating impellers. Like for example, any kind of impellers, if you will see, they will either generate axial flow or they will generate radial flow in the reactor. But there are certain, depending on the blade angle, there are certain impellers which can generate both axial and radial flow. Now, because you need cells suspended in the medium at all the times also, and you need a good homogeneity also, mixing. So, designs like Cetric impellers, marine impellers, although these marine and Ruston turbine are extremes, axial and radial, but in between ones seem to be working well. With plant cell cultivations, people also tend to use large diameter impellers so that there is a good homogenization. Mixing happens at high cell densities. People also use helical blade impellers in plant cell cultivations. So one has to play around with impeller designs. Now, the question which should crop up in the mind is, then again, it is a lot of hit and trial. I keep testing all the impellers, and every batch is taking me a month. So how much time, energy will go into it? What can be a better way to do it? Obviously, if we can have a tool which can predict what will be the impact of the impeller design on the reactor dynamics, hydrodynamics, and it can be made measurable. Although even if I'll come to know that, okay, these will be the shear forces, these will be the gas velocities or the liquid velocities inside, this will be how the liquid, but still it is somewhere ambiguous, because I need to have that quantitative data, and I need to have a relationship what the impact will happen of these changes in the hydrodynamic parameters ultimately on my productivities, isn't it? What I'm looking for is high productivity of biomass, high product productivity. So if in any way I can connect the reactor hydrodynamics with my culture kinetics, then everything becomes measurable to me, easy to gauge in silico. If I tweak this, and because of this, the hydrodynamic parameter changes, so what impact will I see on my biomass growth rates or substrate utilization rates or product formation rates? That is also mathematical modeling. And that is what we also did in one of the studies, which we are about to publish now, where we came up with a computational fluid dynamic based model. And we are also trying to we are trying to make it as a hybrid model to connect it with cellular kinetics, process kinetics, which means trying to connect it with biomass formation rate, substrate utilization rate, and product formation rate. So that in the end, directly at the background, when the simulations are done, I should be able to see that if I use an axial impeller, what impact will it have on my growth kinetics? If I use a helical blade impeller, what impact will it have on my substrate utilization kinetics or growth rate or product formation rates? Cell-to-cell contact can influence secondary metabolism in plant cells. Now, I have been talking about scalability in reactors. So the cells have to be as homogeneous as possible. Otherwise, there will be some scale-up limitations, mass transfer limitations, mixing limitations. But at the same time, I know that the more homogeneous they become, I may have a compromise in secondary metabolism, because there might be a need to have some appreciable amount of cell aggregation. So that has to be kept in mind. That first, so what is important? Before you have come to the reactor part, what were you doing? You were trying to establish the kinetics in the shake flask. You were trying to collect the data. You were trying to understand your culture requirements. So before you even come to the reactor, you must know in secondary metabolism, when you are establishing the kinetics, that whether it is growth associated, non-growth associated, or mixed growth associated, so that you can design, can I have a liberty of a two-stage cultivation? Maybe I need to have one reactor clubbed with another reactor design. Volatile compounds play significant role. So that is about the aeration rate. You may have to optimize aeration rate not only for the oxygen requirements, but if your system, because it is quite species specific, not all plant cells may behave similarly. Now, there may be a chance that volatile components might be very essential for your having a significant impact on your secondary molecule, and maybe a chance where they are not having that significant impact. But all this knowledge you have to gather at the small scale. So when you are doing shake flask cultivations, when you are developing the callus, all these parameters, all this information you should collect. What is the threshold shear for my system? You may work with a herb species. You may also work with a tree species. Both will behave differently. If you are that will also impact the morphology of the in vitro cultures. Sometimes, if you are working with a tree species, you may find they are very slow growing in nature. The in vitro cultures, like for example, we were working with Azadirachta directa indica, neem. Now, the root cultures which we initially developed, hairy root cultures, by agrobacterium mediated transformations, were very thick and brittle. They did not come into the suspension, no growth, when we were trying to develop a suspension culture because of the thickness of the roots. There were oxygen gradients within the root because you could see a tap root and very few branches. It was a tree species. So then we had to go back and search what all factors in the medium impact branching of the roots. Branching of the roots then could be connected to larger number of meristems. The larger number of meristems, which component in the media can be responsible for. Then we figured out from literature that gibberellic acid, somebody has reported, that may give rise to more number of meristems. So then we tried with gibberellic acid in the medium. And you won't believe, the entire morphology changed. We brought it because that is the time when I was doing research. So my batchmate was working with a hairy root cultures on Linum album, which is a shrub. And my project was on Azadirachta indica. So although we successfully had generated hairy roots, we could always see that the Linum album hairy roots have grown, cells has gone to cell suspension, gone to the reactor. They are so happy, the biomass is growing. But with Azadirachta indica, the root was so taproot kind of system, and such small branching happening. I was struggling like anything, to the extent that I used to dream in the night that the shake flask are running on the shaker, and I'm having all the flask, and the hairy roots are growing very well in those flasks. So it has brought so much of stress on my. So then we figured out. So I came across a thesis in literature, a thesis on effect of gibberellic acid. So then we thought of designing experiments and see if gibberellic acid can have a role. And then it completely changed. Now, the root morphology of Azadirachta indica became similar to that of Linum album. And the minute the number of branches increased, the taproot size decreased, the suspension, they started growing in suspension. The mucilage formation, the mass transfer limitations indeed were the key. So that helped. So you need to understand your culture requirements. So everything is not directly extrapolated. So what should be kept in mind when we are doing reactor cultivations? The shear environment. So you need to understand what is the threshold shear for your system. Shear environment, the oxygen transfer capacity, We generally understand the oxygen transfer capacity by a parameter called as volumetric mass transfer coefficient for oxygen. So, oxygen transfer capacity is one. Mixing mechanism. What kind of mixing or impeller should I be using? Then foaming and aggregation. Not all fermentations will show you foaming and aggregation, but with plant cell fermentations, because of the cell aggregation, the cell lysis, which might happen due to the aggregates breaking up, you may be able to observe more amount of foaming, especially in the log phases. So, because of the media composition also, and because of the cell aggregation and the breakage, which may happen. And also, they have a tendency to adhere to the glass walls, plant cells. That's my own experience. You can clearly see that when you are working with plant cell cultures, as they will move towards the log phase, you can see layer of cells building up on the walls. And that is a significant compromise with your overall biomass productivity, which you can achieve. So you need to find a way that, because these are all live cells and freshly growing log phase active cells, if you will not find a way of bringing it back in the medium, they will keep accumulating one on top of the other. And in the end, if you'll allow this, you will observe that the bottommost layer will start becoming necrotic, and it may release components which may further inhibit the growth of the cells in suspension. So one has to find ways. Maybe it requires intermittent change in your aeration pattern or RPM levels to bring those cells, or to look at the ways, maybe addition of baffles to not allow the biomass to settle on the walls. So you need to find ways by which, so baffles will not allow the cells to settle on the walls. So sometimes you need to see what can help to avoid that. Capital investment, that is also important. In the end, whatever design you choose, cost matters. So that is also one of the factors to be kept in mind. And knowing that these are long run fermentations, is the design capable of maintaining sterility for a long time? So modulation of factors like mixing and aeration to balance between the mass transfer requirements and shear sensitivity of the plant cells. So it's a balance, because they are inverse mostly in nature. So if you will try to increase the mixing parameters, there is a good chance that you are compromising on the shear. Now, some of the key properties of plant cell suspensions which should be kept in mind: rheological properties. Plant cell cultures are indeed viscous at high concentrations. As a non-Newtonian fluid, the shear stress, therefore, they are not directly related, stress and strain. Depending on the aggregate size, the cell densities, the viscosity might change. So this will again impact the viscosity and osmolarity, and also then impact further your oxygen mass transfer or mixing. Other things to be kept in mind: plant cell suspensions, they indeed form aggregates. So there can be diffusional limitations. They may become like your immobilized systems. If you will allow the aggregation to keep happening, they'll become like aggregates. So these are nothing like cell immobilized. So what problem you may face is the oxygen gradients which will develop to the inner core. So that may eventually end up compromising with your biomass productivities. Wall growth, I did tell you about this. So to keep the cells in suspension and to provide homogeneous environment is the purpose of mixing, especially with plant cells, because they are high density. So relatively in comparison to microbial cells. So as we have seen, I gave you an example that although they were growing so good, but in the end there was a compromise in the biomass productivity, just because they had become so dense to settle down in the reactor. We were using Cetric impeller, but then the mistake which we did, although now model can help us with that once we noticed that between 8th day and 12th day possibly this is happening, then we could, although after the experiment we realized that there could be a good chance if we had added 2 impellers, which could have improved the mixing. But now again, we'll have to do the experiment. So what was the learning lesson? That maybe in the CFD model which we have developed, in the first hand itself, you anticipate this and see how keeping two impellers at a certain distance on the shaft can improve the hydrodynamics, or can improve, if through the hybrid model, the can, if not improved, at least reproduce the same results as shake flask. So impeller design, shear generation, it is responsible for power requirement. So you must take care of the power requirement. Again, it is a cost. The fluid circulation ability under high viscosity, they can impact, the impeller design can impact. Large diameter impeller can provide high homogenization but lead to less bubble dispersion. So again, a compromise. Although it is a good homogenization efficiency, but at the same time, what are you compromising on? The dispersion of the air. There is a reason why there is a reason why the sparger is just beneath the impeller in your microbial fermenters. Now, the impeller will help you to, otherwise it is flooding, which you will observe in the reactor. They will not disperse in the liquid media, which will eventually end up giving you what? Oxygen mass transfer limitations. All the parts of the reactor will not have enough oxygen dissolved. Baffles are indeed components which are steel components, SS, and they protrude out from the walls. So what happens when during the circulation, when the liquid hits the baffles, there will be small eddies formation which will happen. And these eddies will not allow the biomass to settle on the walls. So in pneumatically agitated vessels, pneumatically agitated means there is no mixing component there. Only air is doing, the bubbling is doing all the mixing. So these are airlift bubble column type reactors. So in pneumatically agitated vessels, but what is the advantage? In pneumatically agitated vessels, these can be good systems if your system on which you are working, the plant cell culture system, is very, very shear sensitive, because they can allow you to cultivate the cell suspensions without having a moving part in the reactor. So there is no moving part, but what compromise will it have? As the density would increase, the mixing requirements have to be met. Now, how to meet those mixing requirements with having no moving part? So people then modified the reactor of bubble column into an airlift system. Airlift system will have a draft tube inside, and depending on the position of the sparger beneath the draft tube, you can have circulation currents built in the reactor. And these liquid circulation currents, which will be built because of the bubbling happening in one direction, then you will be able to have the cells mixed. So this is how mixing can be improved. For the same power per input, your volume mixing time in airlift reactors is lower than in stirred tanks. So, which is a good thing. Mixing, a limiting factor for pneumatically agitated reactors, I just told you. Dissolved oxygen level to be maintained above a critical concentration. So this is a general fact. Whether you are working with microbial fermentations or you are working with plant cell fermentations, you should not allow the dissolved oxygen concentration to go less than some critical value. Now this generally, this critical concentration, as a thumb rule, we say that please don't go less than 10 to 20%. Because there is a good chance that if you'll work on the border line, and if it goes less than the critical oxygen concentration, then your relationship between the specific oxygen demand and the dissolved oxygen concentration is no more, it'll be linearly decreasing. It will not be independent. Okay, so high culture viscosity, cell shear sensitivity, and cell aggregation can make oxygen transfer in plant cell cultures a challenging problem. Talking about shear tolerance varies with species. Please make note, even though I have said that they are relatively shear sensitive, but even among the various species, there can be different tolerance ability. it is important to characterize the hydrodynamic shear for the very reason I told you. If you can bring it in the form of a mathematical equation, you'll have the liberty to see that if I increase the RPM, if I increase the aeration rate, what impact will it have on my shear forces in the reactor? What impact it may have on my KLA, let's say, oxygen transfer coefficient, then what impact it may have on my mixing time, and all these will impact your productivities. These are some schematics. How will the flow look like in pneumatically driven reactors and stirred reactors? The last one, G, is nothing but the rotating drum bioreactor, these are also known configurations for microbial fermentations. So, but these are some of the designs. So you can see a tapered column. So, and these minute changes, some changes are system specific changes which can be brought in if you know your system well. These are different kinds of impellers which you will come across in literature, which are generally used for plant cell cultivations in reactors, stirred reactor systems. So in fact, the position of the sparger in pneumatically driven reactors can play a role in mixing. Like here, an example is given wherein the position of the sparger in the airlift setup, how could it impact the dead zones present in the reactor? Now, once you have finalized the geometrical design, you will do the plant cell cultivations, assuming that you have already optimized at the shake flask level all the parameters. So now you are assuming that you should be able to reproduce in this geometrical design. You'll reach to a productivity. Let's assume you have been able to reproduce the productivities or rather improve the productivities, but all this is in batch. So you generally do batch fermentations. Now, once you have completed the batch, can someone answer me that can there be a way to improve the productivity from batch? Or another way to reframe the question is, what is the limiting factor in batch which is impacting your productivity? Very good. So, one of the solutions to overcome this problem would be what? Even before, in the batch itself. In the batch itself, I'm saying. You said there will be a time where the substrate will be consumed. So, what can be the way out? Okay, that is called as a fill and draw system in batch. Okay, but let's say it is similar. So you will come across many terminologies in literature. People will do some reactor cultivation in batch. Although it has now been opened, you have removed some media and added some, but then people will call it as a batch and will call it as a fill and draw system. I was talking about that once you have realized you had optimized the parameters, which included your initial substrate concentration, the media composition was optimized. You conducted a batch knowing that the limitation would be that, let's say your log phase ends after 12 days. So after 12 days, you have reached to a biomass concentration of X. If I want to improve this X to 2X, what choice do I have in the batch itself? Giving different substrate like diabatic growth or. But I have already optimized my choice of substrate earlier, no? No, I'm talking about the biomass. What will I do as a simple, without having any knowledge? What logically should I do? I'll try to give more substrate, no? So which means, but I cannot open the reactor in between. It's a batch system. So what choice do I have? I'll see if earlier it was S, I'll make it into 2S. Isn't it? Earlier itself, and still conduct the batch, assuming that now that I have given 2S, it will give me better biomass, isn't it? Am I saying wrong? But it may not happen. Although you are expecting, but it may not happen. Why? Why? Because the conditions are S is the substrate is intensity, but all the conditions which is there in the what is this phenomena called? At least those students who are from biotech background. And someone was telling me yesterday that yeah, I was doing BTech biotechnology. We have done courses. At least those people should be able to answer. So there is a term which is called as substrate inhibition. So that is the limitation. So substrate limitation and substrate inhibition, these are the bottlenecks of batch. And what can overcome these bottlenecks is making into a open system. Open system, you have the choice of running it as a fed batch or running it as a continuous cultivation. For high cell densities, people go ahead with fed batch cultivations. So an effective bioreactor operating strategy provides high productivity, high product yield. So generally in fermentations, let me put this very clearly to you people. There should be no confusion. Generally, if you will see microbial fermentations, uh, yield will be with respect to substrate. Whether it is X by S, which is yield of biomass per unit substrate consumed, whether it is product yield, which means the amount of product produced per unit substrate consumed, or it is biomass yield, which means amount of biomass produced per unit substrate consumed. Product yield I have been mentioning in plant cell technology. It means YP by X, which means amount of product produced per unit biomass produced. So generally, this is a terminology used in industry. The yields will be, if I say product yield, it will be product formed per unit substrate consumed. And high product content is amount of product produced per unit biomass. Now, what impact does it have on the bioprocess? A high product content will make your downstream separation and purification easier. High product yield reduces the cost of substrate. Give some time to think about it and let me know if it is clear to you. The first statement was high product content can make your downstream processing easier. Why? Because you are having large amount of product per unit biomass. So possibly the cost of downstream can also be impacted, less reduced. And if the amount of product per unit substrate is also increased, then what am I improving? I'm reducing my production cost. The cost which I'll be spending on the substrates. Because I have improved amount of product produced by unit substrate consumption. Okay. So when we optimize any process, we should work on both the parameters, high product yield and high product content. The operating strategy is determined based on the pattern of product formation. That's very important. So, how do we determine that? From where will you get that knowledge? We had made the cell kinetics in the shake flask that will help you to know whether your product is growth associated or non-growth associated or mixed growth associated. If you'll draw a profile of your biomass or product with respect to time, you can see whether it is in same profile as the biomass or whether your product formation starts once the biomass has reached the end of the log phase and it is continuously increasing. So by this, you will be able to determine whether it is growth associated or non-growth associated. What impact will it have in your designing of bioreactor operating strategy? Let's say you find it non-growth associated. What does that mean? We'll make it more, we'll work it on more batch culture rather than because the more stationary cases, the more. How would you optimize the system? First thing, this you came to know in your shake flask. Shake flask was the place where you were optimizing the media composition, you were optimizing the conditions, also talking about the conditions where the product can be more. So now what does that indicate? That possibly when you are doing all your statistical tools and design of experiments, you will observe yourself that the media components which are impacting the biomass may not be the same as those which are impacting the product. Maybe a nutrient limitation will be increasing your product yield, isn't it? Because that's a stress. Or a particular nutrient might be impacting very highly on your product yield content, but the biomass, it may not be ranking as high, it may be a lower in rank. So if it is completely non-growth associated, ideally you should be thinking about two-stage cultivation, because ultimately productivity matters. So in the first stage, you should implement all the strategies which can improve your biomass, and then switch to conditions which will improve your product inside the biomass. For example, although it is a microbial fermentation example, but nonetheless, like PHP fermentations, polyhydroxybutyrate, your bioplastics. So in Ralstonia, one of the organisms which is known. Now, under nitrogen limitations, the PHB starts getting produced. So then, if you'll see literature, there are two-stage cultivations designed. First, because you need high biomass density, you may have to do fed batch with feeding strategies which will improve the biomass. And then, once you have high cell density, then you switch to feeding those components which will now guide the cells to improve the product. To relate the pattern of product synthesis to cell growth, appropriate index of cell growth is required. So generally in plant cell cultivations, although the phases of the growth remain the same as microbial cell suspensions, you will be able to see a lag, log, stationary, possibly a death phase, but we don't go till that point. Then but how we measure the callus, because callus is being maintained on a solidified medium on plates. So we measure the growth of the callus, which is your master or working cell bank, in terms of growth index. Growth index will be a ratio of the change in the weight of the callus by the initial weight of the callus after certain time, whatever time you have fixed. What are the different modes of cultivations which can happen in the reactor? You can do multi-stage batch, you can do fed batch, you can do multi-stage continuous, you can do repeated batch. That is what somebody was indicating as fill and draw type of a system. Perfusion reactors, which is nothing but your cell recycle. So these terminologies and strategies, they remain the same for any fermentation system, be it microbial system, be it plant cell system, but some optimization of the parameters might be required, which is specific to the culture system on which you are working. Limitations. Plant cell suspension cultures might lose productivity due to their inherent genetic instability. Now, having said all this, suddenly it's important to also tell you that indeed it is easier said than done. There are challenges which do exist because of the very heterogeneous nature of the callus lines. If you are not having a synchronous culture, then you may end up with genetic instabilities. You may end up, which will eventually impact your biochemical activity and your product productivity. Product accumulation. Now, cell suspensions we were talking about, Professor Nathalie also hinted upon that it is compartmentalized, sometimes different tissues, so biosynthetic pathway. You may end up in a scenario where your cell suspension is not showing you any product. It is undetectable. Low yields of secondary metabolite than produced by the parent plants most of the times. The minute you come to in vitro cultures, the untransformed or without optimized wild type, the callus, you will see that the yields are multiple folds lower than what is present in nature. So You'll have to have at least 10 to 20 folds thinking that whatever optimization strategies we propose should be able to at least match up the nature's yield. Although the the good point is that we are always at gain in terms of productivity with respect to time, because in nature it will take a longer time, but in lab it is taking less time. So if you will bring it up numerically in terms of productivities, you may have better values. Low yields of secondary metabolites, I did tell you, expensive hormones required in the growth media. So if one has to work around reducing the production cost, one of the, unlike microbial fermentations or in nature, the cost of the media here is higher. Of course, with animal cell cultures, it is much higher, but nonetheless, if you compare it with microbial fermentations, the cost of the media is higher because of the expensive components like growth hormones present in the media. What is the way out now? How to overcome these challenges? One of the ways by which people have found a way to overcome the said challenges is switching on to organ culture stage. And some of the commercial entities around the world are working with hairy root fermentations now to produce the same compound which the plant is producing. Now, what are hairy root cultures? Yesterday, we were talking about them briefly. Produced by transformation using the natural vector system, Agrobacterium rhizogenes. Although you can end up in a transgenic callus as well, transformed by using Agrobacterium tumefaciens, hairy root cultures is a more stable, genetically stable system, a reliable system for a long drawn industrial application. So therefore, hairy root cultivations have been preferred for those compounds wherein organogenesis is directly related to the expression of the phytomolecule. Now, advantages of hairy root cultures vis-a-vis plant cell suspension cultures. This fast growth is not in comparison of plant cells. Please keep in mind. When we say fast growth, this is relatively in comparison to natural root systems. Now, suppose if you have found that the product is only, the synthesis is only linked to the roots of the system. But if you are trying to generate cell cultures from the aerial parts, you may not be able to see expression of the product. So what one can do is, one can generate hairy roots and they might be able to show the expression of that secondary product. Now, either you generate an adventitious root, a normal root culture, or you develop a hairy root culture. So in comparison to normal root cultures, hairy root cultures are more advantageous in many ways, because hairy root cultures, because of their transformed nature, will not require hormones in the medium to grow, which is a big advantage. They do not require any hormones to multiply, because they have their own endogenous system. Because of this engineering which has happened by the natural bacterium, they are able to independently grow in the suspension. That is a big advantage. Another is, because large number of meristems present, they have better growth rates than normal root cultures. So they have faster growth rates. Genetic stability, for the very fact that they are organ cultures, the problem of soma clonal variation, or if suppose you are using them as a transformant for genetic expression of some gene of interest of yours, then too they are genetically stable systems, because there is integration happening in the genome directly rather than transient expression. So they are more biochemically stable, and higher levels sometimes for the very fact that it has been induced because of the infection by the bacteria. So generally could be related to the defense acceleration. The expression of the secondary metabolites in hairy root systems has been many a times found better than the plant cells. Having said that, there is a challenge. Although now we have been able to overcome the limitations of plant cell suspension cultures, but there is a challenge of scale up because of the very nature of hairy root cultures. See, this is how the hairy roots grow. So hairy roots are biocatalysts, which can have a challenge for scale up. The sheer sensitivity of the hairy root system, because these are organ cultures, is one of the limitations when you are using them in stirred systems. especially. The hydrodynamics will have more impact than on the cells, because the cell size was still less, relatively less than the organ culture. Now the size has increased, the surface area exposed has increased. So there is a good chance or more chance of the impact of shear on them. The breakage can happen, and anywhere the wound gets created, there will be a wound response. The wound response may manifest in the formation of callus on those regions. So you may observe variation in the yields. Requirement of support matrix. I did mention that they are self-immobilizing in nature. Self-immobilizing in that to kickstart further branching or multiplication, the roots require a support matrix, self-immobilize. And this is facilitated if they are inoculated onto a support. Now, we ourselves had faced that challenge when we were working with Azadirachta indica hairy roots, when we were bringing it into reactor systems, when we were using stirred reactors and pneumatically driven reactors. Although everything was working well in the shake flask, the conditions were optimized. 25 days was the cultivation time. When we brought it into the reactor, for 25 days, the root kept on dissolved in the reactors, but by the end of the 25th day, it was still as they looked on day 0. No growth. We did the spent media analysis and we found that the carbon was still there where it was on day 0, which means they were not even taking up carbon. The growth did not happen. What can be the reason? We tried to change the configuration. We brought them into pneumatically driven. I told you, first stirred and then pneumatically driven, still the same. No change. Now, then we thought that is self-immobilization, because in shake flask, then we started comparing why they were growing in shake flask. Then we were thinking, when we reanalyzed the data, that initially when we were developing cell suspension cultures, the media, the flask volume to medium volume ratio was impacting the growth. So what we realized that when the this flask volume to volume ratio was being optimized, the roots before beginning, the lag phase was longer, which was around 4 to 5 days. Before start multiplying, they were showing accumulation. And only when they had accumulated to a point, then the fresh roots were coming up. Further multiplication was happening, which was not happening in the reactor because the reactor hydrodynamics were not allowing it to come together. So then we thought that possibly there is a need for support for the roots. So then we added and modified the reactor design and we inoculated the roots on support. We used a polyurethane foam support where we inoculated the roots and we floated it onto the reactor. And we saw that it started growing. There were other factors also which we kept in mind, the hyperhydricity, which was also impacting the hairy root cultivations, a lot of water retention on the roots. They were not liking it. So we also looked at mist bioreactor designs, wherein there is less of media retention on the surface of the roots, because possibly large hyperhydricity was leading to large amount of thick boundary layers around the hairy root surface. So because of this, all boundary layer problems and phenomena were cropping up, the mass transfer limitation, especially the critical factor was oxygen mass transfer limitation to them. So what may happen, depending on the morphology of the roots, you may also end up looking at oxygen gradients. It's not easy to look through the eyes that, oh, possibly this could be the reason. So when we were working with hairy roots with Azadirachta indica, one was what we identified, they need support. Now, once they started growing, we saw that it was not matching the shake flask productivity. What else can be the reason? So when we were trying to increase the volume, so then we did spent media analysis. We found that no, it is taking up carbon, there is growth, but still then after 25 days, still the carbon is left. So there is some other limitation. It's not the limitation of carbon because carbon was there in the medium. So there is something else which is now causing a limitation. So then we thought that can there be oxygen mass transfer limitation because of the very nature of the roots matrix. So then we thought that let's increase the aeration rate and try to see. So at the small volume, we then increase the aeration, the sparging rate into this. And we saw that it did improve, but the improvement was not that appreciable. Then we thought that, okay, air sparging rate improvement is not also giving us much respite. So what should we do? So then we thought, is it specifically oxygen limitation to the inner core? Although we are giving it in the dissolved. So now we were also simultaneously checking the dissolved oxygen level in the media which was present, and it was 100% saturation. So then we were thinking that the media around the roots is showing bulk oxygen concentration of 100%, and we were sparging in air. Still, the roots are not able to take up carbon beyond a point, and they are not growing. What can be the reason? So then we thought that let's try and mix up pure oxygen with air and see if it is the oxygen which is the sparingly soluble component in the medium, despite the fact that the aeration rates have improved, but the oxygen is not able to reach in the required quantities to the inner core of the matrix. So then when we started mixing and improving pure oxygen with air in that small volume, oh, they became so happy. They flourished like anything, which gave us an indication that and it matched. And sometimes in some cases, I think with 100% oxygen, it went beyond shake flask. So that told us what? That it is oxygen mass transfer which is a limiting factor than generally observed in fermentations, your limiting substrate in the medium which was carbon or nitrogen. So then we figured out for organ cultures like hairy root cultures, possibly the scale up criteria should be based on oxygen transport rate, or rather than working around with your medium nutrients limitation. So first bottleneck was removed when we worked around the oxygen mass transfer. The minute we gave them oxygen enough, the carbon started getting utilized for their growth, and we saw improvement. So it it looks very straightforward in books. When you'll read, when you'll do bioreactor design and analysis course, the concepts will be there, but you need to work around multiple factors. You need to be passionate about your system. You need to really think what can be the reason methodically, systematically, and design your experiments accordingly. So it's a balancing act between the biological needs of the tissue without inducing any additional or undesirable biological response. Now, when I said, especially in case of organ cultures, when it will grow like a ball, rough ball with all mesh. Now, oxygen will be one of the critical components. If you'll, after harvesting, if you'll open up that ball, you will see that inside core is dead, necrotic. So if you can find a way of during the entire cultivation, the oxygen to keep flowing in, there are no limitations happening till the inner core, you may be able to improve your biomass productivities and product productivities. So what can be done? Now, how to scale up and find the right reactor designs for such systems. So if you'll go back to literature, you'll find two ways. One is liquid phase reactors, and the second classification is gas phase reactors, especially for such cultures, organ cultures. People have worked around because liquid fermentations are easier to scale, easier to control. So people begin with liquid fermentations. So in liquid phase reactors, how can you manipulate or bring in all these requirements? If you see reactors available for hairy root cultivations, people have used stirred reactor systems, but with a modification. Knowing that they are self-immobilizing, the impeller has been at the bottom. They create a division such that the roots are not directly exposed to the impeller, and the roots are also given a support. So they'll create a mesh-like structure. I'll show you the picture here. So you can see that A picture. So wherein the roots will be inoculated. So the self-immobilizing nature is facilitated. The mixing is facilitated by the bottom part of the reactor where the mixer is there. Or people are using bubble column reactors if they are finding it highly shear sensitive. But at the same time, I told you now, because they are self-immobilizing in nature, there's a good chance that as they would grow, the inner core will become mass transfer limited. So you may not end up in having what you were expecting as the biomass productivity. What is the way out? The way out is that when you are initially inoculating, you divide the inoculum into much smaller parts. Let's say if you are working around with 10 grams per liter of inoculum, I'm giving you a scenario. Why don't you divide the inoculum in the same reactor into 2 into 5? So which means have a support for 2 grams, another support for 2 grams. And then people have come up with designs where they have segmented the reactor. And so various designs to meet the culture requirements, you will see, have come across in literature. Then people have also planned to divide. I was talking about the hyperhydricity problem with organ cultures or root cultures, which means a lot of water retention can lead to thicker boundary layers and therefore nutrient and mass transfer limitations. So how can you avoid that problem? You have to reduce the boundary layer phenomena, which means you do not allow the water retention to happen on the roots. So what can you do? So people have in liquid fermentations divided into two chambers. The culture chambers has been separated from the mixing tank, and it has now become a trickle bed reactor where you inoculate the organs, the hairy roots, and you keep recirculating your media components by spraying onto the roots, and then it goes back to the mixing tank. We again aerate it to fulfill the oxygen, dissolved oxygen there, and you keep recirculating. I also wanted to say, despite all this, sometimes you may end up in certain limitations in liquid phase reactors. What are the limitations? As the root density would increase, the pressure drop across the reactor can limit the mass transport. Although in that reactor, I told you they have segmented into two parts. One has now become a packed column with the hairy roots. But as the hairy roots would grow, the pressure drop would grow across this column. So it will cause hindrance on the flow and sometimes mass transfer limitations even within the growing hairy roots. That is one limitation which may happen in such systems as well. The extra aeration required to meet the oxygen demand of the growing roots is challenged to achieve in liquid reactors. Mucilage present on the roots growing completely suspended in the liquid or with large amount of liquid to air ratio will also lead to resistance in oxygen transfer. Root hairs decrease mass transfer and oxygen availability. Because these are hairy roots, you'll observe how they are different from normal roots in various ways. They are plagiotropic, not geotropic, and they will show large amount of small root hairs on their branches, more than what you can see in normal root systems. So now every such root hair is also adding on to the mass transfer limitation. So then what is the choice? People came up with modified reactor designs called as gas phase reactor designs. How are they different from liquid phase reactor designs? In liquid phase reactor designs, liquid is the continuous phase and gas is the dispersed phase, isn't it? Now in gas phase reactors, gas becomes the continuous phase and liquid becomes the dispersed phase. So the reactors in which liquid is the dispersed phase and gas is the continuous phase, the roots are exposed to air more now, isn't it? More air means why liquid fermentations oxygen is a limiting factor. Why? Anybody? So it is the most sparingly soluble component of the fermentation medium, isn't it? Most of the times. And therefore, oxygen transfer is one of the key points which have to be looked into when you are working around fermentation optimization. Eliminating any oxygen deficiency in dense root beds. Now, what has happened? You have air, lesser water, so boundary layers are less. OTR is what? Oxygen transport rate. If there is higher gradient, what is the driving force for higher oxygen transfer? Concentration? Gradient. That is the driving force. Oxygen present in the air is in much higher concentrations than what it was in your liquid media. Now the gradient is more and therefore more oxygen transport rate. Low shear stress environment, there are no moving parts. no hydrodynamic shear, so lesser shear environment. Complete control of gases. That's obvious because we have less water activity. So what kind of designs you will come across in literature for hairy root cultivations? Nutrient mist bioreactors, trickle bed bioreactors, radial flow reactors, and many more. Droplet. So now one of the most popular design is mist bioreactors, which you will come across in gas phase reactors. We ourselves also worked on it, where people manipulate the size of the liquid droplet. So people use ultrasonic transducers to convert the liquid media to a cloud form. So what all factors have to be optimized? The rate at which, the cycle time. How many times are you exposing the media droplets to the biomass in one go per unit time? Then what other factor? The size of the droplet. So maybe a mist size, which is a nano, possibly a nano nozzle might be required, nano size, which can give you a very small size of media droplet, which can easily come and settle, or like a cloud. So all these factors have found to play a role in your biomass. We ourselves, when we were working on reactor cultivations for Aza directa, indica, hairy roots, and optimizing the reactor design, we worked on trickle bed reactors where we were trying to reduce the size of the droplet. We also worked on mist bioreactors, where in one go we tried to use an ultrasonic transducer to convert the media into clouds and then allow it to get exposed to the culture chamber. In the other case, we used a a nozzle which was determined to give us a nano sized droplets. So, and best results were achieved in mist bioreactors. So, because possibly mass transfer limitations and oxygen mass transfer limitations were the limiting factor for the hairy roots in a reactor system. There are other designs available for plant cell organ cultures. What you can see in literature is for micropropagation or for embryogenesis, people have come up with bubble. These are called as balloon type reactors. So they don't look similar to what you conventionally see as reactors. But again, scalability can be an issue. But what people have also come up as an alternative is, rather than doing a large bioreactor in production, like 200,000 liter reactor, you can have multiple batches with a smaller size. So people like Rutec Bioactives, they have come up with a series of reactors in one line, wherein the size of the reactors have been reduced, and they are working with hairy root cultures with a smaller batch size of the reactors. See, this is how an embryonic cell culture has been done. This is used for micropropagation. So you can use the reactor systems not only to generate biomass for extracting out phytochemicals, but this can also have an agricultural application. If you are working with micropropagation, then possibly you can use these reactors in one go to give you thousands of embryos in just 1 liter or 500 ml of your suspension. And each of these somatic embryos can lead to one single plant. So these are some of the examples where people have done micropropagation for various plant species in these balloon type reactors. And this is mainly an in vitro cultivation of whole plants. If you're working on a nursery and you want to do hardening and then do mass multiplication in the field, then possibly the reactors have a role to play there as well to speed up the cultivation. Okay, this is an example where people have tried to produce cyclotides by using cell suspension cultures, and they have come up with their own reactor design which has a very high aspect ratio. When I say aspect ratio, it is height to diameter ratio. They will look like a long bubble column and an airlift reactor, where because they were wanting to do photo systems. Okay, that's it.